Good morning, everyone. It's my pleasure to present our study, Gut Microbiome Metabolite-Target Access in Parkinson Disease. The main idea is simple. We wanted to move from gut microbiome metabolites to testable host target access. So we asked, which microbiome metabolites may interact with which host targets in PD? Parkinson disease is a progressive neurodegenerative disease. It's marked by dopaminergic neural loss and motor symptoms. But PD is not only a brain disease. Many patients also have non-motor symptoms. Gastrointestinal symptoms such as constipation may appear before motor symptoms. PD is also related to alpha-synuclein aggregation and neuroinflammation. This point supports the idea of a gut-brain exercise. In this context, signals from the gut may help us understand PD-related mechanisms. The gut microbiota can affect the host in many ways. One important way is through some molecules made or changed by gut microbes. These molecules are called microbial metabolites. Microbiome metabolites may affect the intestinal barrier, immune responses, the blood-brain barrier, and central inflammation. Bacterial composition is useful information, but metabolites may give us a more direct mechanism. For example, a metabolite can be linked to a host target and then to a pathway. So, in this study, we focus on microbiome metabolites as possible molecular links between the gut microbiota and PD-related host pathways. Many studies have reported gut microbiome changed in PD, but some gaps remain. First, many studies stay at the level of broad microbiome association or broad inflammation. Second, hub genes can show important host genes, but they may not tell us where the upstream microbiome signal comes from. Therefore, our aim was to prioritize traceable gut microbiome metabolite target access in PD. The output was not proof of treatment effect. It was a set of candidate access for future validation. The point is important. Our study should be viewed as a computational prioritization study. It gives hypotheses. It doesn't replace experimental validation. The slide showed our workflow. First, we collected gut microbiome metabolites. Then we predict their possible host targets. At the same time, we collected PD-associated genes from disease databases. Next, we found the common targets between the metabolite-related target and the PD-related targets. Then we used PPI analysis and functional enrichment analysis. After that, we built a Microbiota-Substrate-Metabolite-Target network. We called it the MSMT network. This network helped us trace the source of each candidate axis. Finally, we used admit profiling, molecular docking, and 100 nanosecond molecular dynamics simulation. In short, the workflow moved from target screening to work analysis and to network analysis to source tracing and then to structure and dynamic evaluation. We first integrated metabolite-related target with PD-associated targets. From the microbiome metabolite side, we obtained 1,521 predict targets. From the PD side, we collected 2,667 PD-associated targets. After intersection, 54 common key targets were returned. These 54 targets may connect gut microbiome metabolites with PD-related host biology. The network also shows several important themes. These included inflammation, oxidative stress, cell survival, and cell death. These themes are all relevant to PD. At this stage, we didn't assume that all 54 targets were equally important. Instead, this target set was used for the next step of network prioritization. Next, we performed PPI analysis. The PPI contained 54 nodes and 643 edges. This means the target formed a connected network. Based on topical ranking, 5 core targets were selected: AKT1, IL-6, TNF, TP53, and IL-1 beta. This target was meaningful for PD biology. AKT1 is related to cell survival. IL-6, TNF, and IL-1 beta are important inflammatory mediators. TP53 is related to stress response and cell bias. Together, these targets connect inflammation, survival signaling, stress response, and cell death. This also helped us reduce the complexity of the study, so we use this 5 target as a main host target layer for later analysis. We then performed functional enrichment analysis. For GO analysis, the enriched terms include response to bacteria moleculars, response to lipopolysaccharide, reactive oxygen species metabolism, and regulation of apoptosis. These terms are related to the gut immune-brain interface. For KEGG, several pathways were enriched. These include toll-like receptor, PI3K, AKT, MAPK, TNF, IL-17, and node-like receptor signaling. These pathways are related to immune activation, inflammation, cell survival, and cell fate. So the results were consistent with our biological question. This result supports biology plausibility, but it doesn't prove clarity. It only gives pathway context for the next steps. Next, we evaluated candidate metabolites using adenine profiling. The purpose was to check whether the metabolites had accessible predict drug likeness and safety-related features. As shown in the heat map, the top candidates show generally favorable profiles. One compound, 2-hydroxy 3,4-hydroxyphenyl propanoic acid, ranked highly. Also, no top 10 metabolites violated Lipski's rule of 5. This made this compound suitable for further computational screening. Again, this doesn't mean these metabolites are ready as drug. It's only a fleeting step before structure and dynamic evaluation. A key part of our study was the MSMT network. This network includes 69 microbes, 23 substrates, 31 metabolites, 5 core targets, and 158 edits. The main purpose was source traceability. In many computational studies, we can see a compound-target relation, but the upstream microbiome context is often unclear. Here, we linked microbes, substrates, metabolites, and host target in one network. This helped us make the candidate access easier to interpret. It also made the results more useful for future experiments. For example, researchers can examine microbiome producers, substrate available, metabolite levels, and target responses. So the MSMT network connected prediction result with possible biology follow-up. And next, docking showed possible binding poses between metabolites and target. From the docking result, we focused on three representative axes. The first was 2-hydroxy 3,4-hydroxyphenyl propanoic acid RL1 beta. The second was interradial RL6. The third was indole 3-lactic acid AKT1. Based on source training, ADMET profiling, and docking, we selected 3 axes for 100 nanosecond MD simulation. All these 3 axes were traceable in the MSMT network. They also have favorable ADMET profiles and plausible docking poses. But these results were not enough to test dynamic stability, so we used MD simulation to see how the complex behaved over time. We look at several indicators. Protein RMSD and ligand RMSD show structure moment. RG reflects compactness. FEL helps show stable conformational states. This step helps us compare the 3 axes and separate a leading candidate from secondary candidates. Among the 3 axes, 2-hydroxy 3,4-hydroxyphenyl propanoic acid IL-1 beta shows the most balanced and dynamic behavior. First, the protein RMSD became stable after the early part of the simulation. This suggests that the protein reached a relatively stable state. Second, the ligand RMSD stayed within a moderate range. It didn't show persistent dissociation from the binding environment. Third, the RG remained relatively narrow. It suggests that the protein kept its overall compactness during the 100 nanosecond simulation. The FEL also showed connected low energy basins. They support relatively stable conformational states. Taken together, these complexes had stronger dynamics support than the other 2 axes. Therefore, we prioritized 2-hydroxy 3,4-hydroxyphenyl propanoic acid IL-1 beta as the leading candidate axis. But this is still computational evidence. It doesn't prove direct biology inhibition or treatment effect. It only suggests that this axis is worth testing in future experiments. The other 2 axes was interpreted as secondary candidates. For interradial IL-6, docking suggested a favorable pose. But during MD simulation, ligand RMSD showed clear spikes. This may suggest transient ligand drift or weaker ligand retention. So the docking result was good, but the dynamic result was not equally strong. For indole-3 lactic acid AKT1, the axis is biologically reasonable because AKT1 is related to survival and inflammation-related signaling. However, its ligand trajectory also showed repeated fluctuation. Therefore, we didn't treat these two axes as equal to the primary axis. We considered them secondary candidate axes that still need further validation. This cautious interpretation is important for computational studies. It helps avoid overclaiming and make the conclusion more balanced. So, in conclusion, this study built an integrated computational framework for PD. The framework helped us prioritize traceable gut microbiome metabolite host target axis. The study moved from broad microbiome association to the specific metabolite target axis. The leading candidate axis was 2-hydroxy-3,4-hydroxyphenyl propanoic acid IL-1 beta, interradial IL-6, and indole-3 lactic acid AKT1 remained secondary candidates. Future work should include repeated MD simulation, binding free energy analysis, affinity assays, cell models, and animal models. These experiments are needed before any strong biological or therapeutic conclusion can be made. Overall, our study provides focused and testable hypotheses for studying gut microbiome metabolites and host inflammatory interface in PD. Thank you very much for your attention. I would be happy to take any question.